I'm here today to talk about a lesser known fact but a well-accepted hallmark of cancer is that there are abnormal sugar coats seen on the surface of cancer cells, but not seen on normal cells. And I think with the cell therapy and engineering era that we're currently in, we now have the technology to actually target these abnormal cells to help patients with a variety of different metastatic solid tumors. So that's the basis of my research. So metastatic disease, which is cancer that's spread to distant organs, is responsible for 90% of cancer deaths. And so while we've gained a better appreciation of how tumors progress, prognoses for patients with advanced disease still remains very poor. And so everybody in this room knows somebody who's been impacted with cancer. And so this is a very strong common denominator that unites all of us. And so an estimated 10 million people worldwide are expected to succumb to this disease annually. And so this piece of data coming from the US Seer Cancer Registry highlights the estimated cancer deaths in 2024. And as we can see, the majority of those cancer deaths are from solid tumors. So we still need better treatments for these. We do not have cures for a lot of metastatic solid tumors. And every time I'm in the clinic, I have to have at least one conversation with patient and their families that we have run out of standard of care treatments for them. So there's a glaring unmet need for us to develop new, better, effective anti-tumor therapies for patients with metastatic disease. And so my overall goal is to be able to cure specifically metastases so that patients can live longer and have better lives. The current standard of care for solid tumors largely is focused on systemic therapy for advanced disease. So this is usually a combination of chemotherapy with immunotherapy plus minus targeted therapies. And so a major revolution in oncology is the advent of the immunotherapy era, which we're still living in right now. The most commonly employed class of immunotherapies is checkpoint inhibitors. And these work largely by broadly interfering with the molecular breaks seen in immune cells, mostly T cells, but also other types of immune cells. And this enables one's own immune system to fight against the cancer. And so while checkpoint inhibitors have improved overall survival for a variety of solid different tumors, it only works for a minority of patients. And this is thought due to a lack of specificity to the tumors. And there's limitations due to toxicities of overactivation of the immune response against normal healthy tissues, leading to autoimmune pathologies. In contrast, cell therapies are living drugs that can be specifically engineered to recognize tumors. And so this is something, one single infusion of a cell therapy can actually last for many years and even cure patients that we've seen through a process called memory. This is kind of a schematic of the general gist, and this is focused on blood cancer, but we take the immune cells out from a patient. We can introduce an engineered receptor that can specifically recognize a tumor protein, expand them, and then reintroduce them back into the patient. And this has been shown to cure actually several refractory blood cancers. And so there's now 7 FDA-approved cell therapy products. 5 of them are focused more on the blood cancers, and now there's 2 that have just been passed this past year on melanoma and sarcomas. And so there's growing interest that we can advance this type of therapy for solid tumors. But overall, engineered cell therapies for solid tumors have remained largely elusive. And the reason why there has not been the great successes that we see in blood cancers is that a universal tumor target is not really been able to be found that's distinct from healthy tissue. So this brings me to my research, where on the cell surface there exists a variety of complex, elongated chains of sugars on cells, and this decorates lipids, proteins, and even nucleic acids, and these are critical for day-to-day function. And so these sugars are also called carbohydrates and glycans, and so all cells have them, and they look like, if you think of a tree with a sugar core, and then long chains of different types of sugars as the branches, and so that's what's seen on a normal healthy cell. The sugar cores are typically masked in normal cells, but become unmasked upon malignant transformation into cancer. And this has been shown to help the tumor evade the immune system, get into the blood vessels, invade tissues, and spread into distant organs. And so typical therapeutic targets are typically focused on highly abundant proteins on the surface of the cell, such as HER2, but these sugar cores are actually 10 to 100-fold higher on the surface of the cell, so they could be seen as optimal targets. But even though this well understood and accepted biology, there's still, it's been difficult to raise classical antibodies used for developmental therapeutics that specifically target these sugar cores. And so in particular, the O-type sugar cores that have been found on a range of different types of solid tumors, such as, not limited, to breast, prostate, gastric, ovarian, pancreatic, and lung cancers, provide kind of an ideal target that we currently don't have good treatments for. So I've developed a T-cell therapy using a novel receptor shown by the green cell that's able to bind specifically to the O-type truncated sugar cores that's seen on tumor cells, as seen by the red cell, and demonstrate functional cell killing in a petri dish and in a mouse model of lung cancer. Additionally, through cell staining of human tissue microarrays, I'm able to show that this novel receptor is able to bind to tumors, only tumor tissues and not on normal primary tissue. And we've also shown through flow cytometry and tissue staining that this particular sugar core is highly expressed on distant metastases in comparison to the primary tumor itself. So I'm working towards meeting the required benchmarks to hopefully bring this therapy into the clinic to benefit patients. So I believe that these sugars that are only seen on cancer offer ideal targets for a new class of cell therapies capable of eradicating metastatic disease.